it's important to know where the cells are coming from. So this is a diagram showing that pluripotent stem cell can go two different ways, and by two different ways I mean we can have a lymphoid stem cell achieved from here or myeloid stem cell. After lymphoid cell is produced, it may transform to natural killer progenitor cell, T cell progenitor, and B cell progenitor. So natural killer progenitor eventually will turn in natural killer real cell. T cells will be transformed through time as into a T cell, T cell progenitor. B cell will mature into plasma cell. Unlike lymphoid stem cells, myeloid stem cells give origin to many different cell types and to majority of the cells you'll find in circulation. So at this point, myeloid stem cell can break down to monocytes, which will be turn monocyte progenitors, we can say, which will turn into monoblast and then monocytes. Granulocytes, this is an important concept, granulocytes can be transformed into three different types of cells, which are also considered granulocytes. So they can be eosinophils, neutrophils, basophils. At the same time, they can be considered still granulocytes because they have the granules. And as we talked before, granules can have substances inside them that may have some toxic properties for the invading organism. Megakaryocyte, it will be transferring into platelets, and erythrocyte progenitor cell will transform to reticulocyte, which is immature cell, immature red blood cells, which will turn into erythrocyte. Erythrocyte, of course, we know already that it doesn't have a nucleus. Instead of it, it has a indentation which makes it look like a donut with no hole. And again, the indentation signifies the place where nucleus used to be. White blood cells can be named also leukocytes. And again, one of the important part or components of white blood cells are granulocytes. Again, the name granulocytes comes from the notion that they have granules inside. Neutrophils, eosinophils, and basophils. So neutrophils are primary pathogen fighting cells. Neutrophils are the first responders to an invasion. Eosinophils, they will help control allergic responses and fight parasites. Also, they involve into processes such as asthma. So please remember the expression again: worms, wheezes, and weird diseases. So these are the eosinophils, what they do. Basophils release heparin, histamine, and other inflammatory mediators. And basophils, we talked about it in the past, that they can be transformed into mast cells when they migrate inside of the tissues. But again, these all three types of cells have uniting feature or common feature, which is presence of granules. And granules have some type of toxic or inflammatory mediator containing granules, and the granules make them different. When we stain those cells under microscope, we will differentiate them from granulocytes, which do not have these inclusions, which do not have these granules. Lymphocytes, it's the different type of cell. So we have B cells that create antibodies and T cells that control the immune system. B cells create antibodies and they produce antibodies, and T cells will be responsible for cell-mediated immunity. So lymphocytes coming from the lymphoid stem cells, and of course they can be found in the bone marrow. Lymphocytes constitute approximately 30% of white blood cell counts. Sometimes it can be lower, but usually the number we like to see is 30. Again, there are no granules, so because of that we can call them a granulocyte. So it's B cells, T cells, and natural killer cells. So B lymphocytes, they are antibody producing cells, as I said, and they are involved in humoral mediated immunity. The second type of cell, as we said, was T lymphocytes, and they will create cell-mediated immunity. Natural killer cells are lymphocytes who have different receptors than, let's say, B cells and T cells, and they naturally kill. So natural killer cells are a somewhat different type cells from B and T cells. So how we distinguish all these three types of cells? The three types of cells or three types of lymphocytes are distinguished by their surface molecules, and by molecules we mean markers, and which certainly can be identified by use of advanced methods, and we can use something like monoclonal antibodies. But to go back to the markers or molecules on the surface, these are molecules are clusters of differentiations, and they can be grouped into categories that are specifics for cells of different origin. So let's say helper T cells, they will have CD4 markers, cluster of differentiation marker, and the effector T cell CD8 marker, and these are like really simple examples of they always brought up, but CD markers are widely used to identify the cells. So again, the cells really may look the same, more or less, but what makes them different is the markers on the surface of the cells. So monocytes are the biggest cells in white blood cells. So they are approximately 6%. So 6%, and when we say 6% about monocytes or other white or even red blood cells, there is no set number, of course. It's a median of percentage of monocytes, and the number will fluctuate, and between 3 and 8% this will be normal count. But again, it's easy to remember 6% for monocytes. So monocytes, they are the largest, and they have U shape. So, and the monocytes, they have relatively long lifespan, and it's approximately 1 to 3 days. Of course, compared to red cells, it's nothing, red cell lifespan, but however, compared to, let's say, lifespan of neutrophils, it's approximately 4 times longer, and it helps them to function longer and produce better results, let's say. So the cells also can survive in the tissues for very long time, for several months to years. So when macrophage or when monocytes turns into the cell, into the tissue, it turns into macrophage. Again, when monocyte migrates into the cell, it's labeled as the macrophage. So when it enters into the tissues. So again, they have very distinct cell roles. They may be cells that play a role in inflammation, as well as they have antigen presenting roles to cells. So these are the cells that actually will digest the invader and but will present it at the same time to the T cells. So the role of monocytes, macrophages, is important because they are presenting cells, and it's somewhat a different type of cell from granulocytes and agranulocytes because of their long survival rate and their ability to present antigens to T cells. This is a picture that gives you some kind of notion to understand what's happening with the cells and how they look. You can see the granulocytes, neutrophils, eosinophils, and basophils, distinct granules. Think about granules as lysosomes, and lysosomes that have some kind of substance, again, toxic to the invaders. A granulocyte, lymphocyte, has distinct nucleus, but we don't see observable granules. And monocytes are having distinct U-shape, and by distinct U-shape, I mean by distinct U-shape that they have U-shape themselves. Their nucleus have a U-shape, but this is depiction of different types of cells. On the background you can see these pale circles, and there are the erythrocytes that do not really define, that are not defined on this picture from the microscope. So this is another depiction of transformation of myeloid stem cell already into end results: platelets, monocytes and granulocytes, and erythrocytes. So what happened here? This cell actually comes from pluripotent stem cell, and it will turn into committed precursor cell. By committed precursor cell, we mean colony forming units. The colony forming units, they would be precursor cells that will eventually turn into the type of cell that we need in circulation. This process doesn't happen by itself. For the transformation to occur, we need colony stimulating factors. Colony stimulator factors are chemical substances that are affecting bone marrow to produce certain type of cells. The most common and most known one is erythropoietin, it's stimulating factor that will affect bone marrow to produce erythrocytes. It's also available in synthetic form as a pharmaceutical to encourage people with some type of bone marrow suppression or blood loss secondary to one or the other factor, let's say radiation, chemotherapy. We can give this and encourage the bone marrow to produce erythrocytes. But also there are other colony stimulating factors such as thrombopoietin, which will result in which will encourage or facilitate platelet production. So on the other hand, what else can happen to pluripotent stem cells? It may turn into lymphoid stem cells. So from lymphoid stem cells it will turn into committed precursor cells for T cell progenitor or B cell progenitor. And T cell progenitor and B cell progenitor will turn into mature cells. Well, T cell progenitor will work through thymus and they eventually will turn into T cells, and B cells will become plasma cells. Okay, and also natural killer cells will occur from this process. So this is another depiction of the entire process, and this diagram will show you where the cells are coming from. This diagram is important to imagine or to have a understanding of what happens with cell lines when one or the other cell malignancy occurs. So again, this is the same slide. So what happens if we're having white blood cells deficiencies? We can have leukopenia, deficiency of leukocytes in general. We can have neutropenia, and by neutropenia usually the general umbrella term is agranulocytosis, because we really cannot differentiate in, especially in old-fashioned lab technique when we were using the basically the microscope and the lab technician's eyes to differentiate between types of granulocytes. So the general term will be agranulocytosis, but it would mean most of the time that we are lacking neutrophils, also eosinophils and basophils. Aplastic anemia, infectious mononucleosis are other sources for the white blood cell deficiencies to occur. So not only white blood cell disorders can affect the count of white blood cells, but something that may affect other instances such as infectious disease, viral disease, or even anemia.